as we discussed earlier, proteins are very important targets for drugs, and a large number of drugs interact with proteins. We also looked at what is the structure of a protein, and in some detail we discussed about what are the various aspects of a structure of a protein. Right now, we look at how proteins function. So, before we go there, let us look at what are the different types of proteins and how they are classified. So, the first class of protein that we shall look at are structural proteins, and these are proteins which help, as the name suggests, help with maintaining the structure of the cell. So, an example here is tubulin. So, tubulin is a very important protein because it can undergo polymerization and depolymerization. Okay, so, it provides a very important structure of the spindle during cell division. Okay. So, here in this case a parent cell divides into two daughter cells, and the spindle formation is a really important part of cell division. So, during cell division, tubulin undergoes polymerization, and then therefore, it forms a structure called spindle, and these actually form a framework on which the chromosomes of the original cell are transferred to the nuclei of the daughter cells. Okay. So, a disease such as cancer, the cells are rapidly dividing, and so therefore, tubulin polymerization must occur at a very high rate, and targeting these structural proteins can be useful in the development of cancer drugs. The next class of proteins that are important are transport proteins. Okay. Transport proteins occur on the surface of cells, and they help in carrying polar molecules such as small amino acids, sugars or nucleobases across the cell membrane, and now the cell can use this for the synthesis of proteins, carbohydrates and nucleic acids. And they are also very important in transporting neurotransmitters back into the neuron, okay, after the signal has been transmitted. Okay. We will look in shortly why this is important. So, why is this smuggling operation necessary? Okay. The question is, why can't they go through on their own across the cell membrane, right? As we looked at earlier, the structure of a cell has a lipid bilayer as a barrier or as a membrane. The lipid bilayer forms an important structure that acts as a barrier in a cell, and these lipids are hydrophobic in nature. And so, therefore, having a hydrophilic molecule with polar surfaces and polar structures, it is very difficult for these to cross the hydrophobic cell membrane. And if we do not have a mechanism by which these polar structures are taken in, then it becomes impossible for us to synthesize proteins, nucleic acids or carbohydrates, which are all important cellular components. Okay. So, let us look at how this transport protein typically functions. So, the transport protein has a area or the surface which is actually hydrophobic, contrary to how typical proteins are which we just learned. How typical proteins fold, these actually have hydrophobic surfaces, and their internal residues are actually hydrophilic. Okay. And this hydrophilic residues interact with the polar molecule and they can bind it, right. Once they bind, it induces a conformational change and it gets entrapped in the protein, and now again another series of conformational changes can occur through which the protein is actually kicking out the polar molecule. Okay. So, it is almost like how you can stow away a small molecule in a pocket and ferry it across the membrane to release it on the other side. So, this is how typically a transport protein works. Now, transport proteins are important targets for several drugs, and we shall look into these in detail later. Okay. Now, the most important proteins other than structural proteins and transport proteins are enzymes and receptors. Okay. So, now let us spend a little bit of time and look at what enzymes are. Enzymes are natural proteins that can catalyze chemical reactions. Okay. Of course, ribonucleic acids, which is also called as RNA, can also catalyze reactions, but a vast majority of the catalytic processes are mediated by proteins, which are called as enzymes. The first enzyme to be recognized as a protein was a urease, which was isolated from jack bean, and it was crystallized in 1926 by Sumner. Okay. James Sumner subsequently won the Nobel Prize in 1946 for this discovery, and this enzyme catalyzes the reaction of urea to carbon dioxide and ammonia. By the 1950s, there were hundreds of enzymes that had been discovered, and many were purified and also crystallized. Okay. In 1960, the first sequence of an enzyme was discovered by these three scientists, and this was of ribonuclease A, which had only 124 amino acids, okay, and the molecular weight was around 14,000 Dalton. And this is a really elegant piece of work, and Stein and Moore shared a Nobel Prize in 1972 for discovering the methodology of protein sequencing. Okay. And so, therefore, by the 1950s and 60s, people had not only isolated a protein, but also were able to sequence it and identify what the sequence was. Right. Subsequently, enzymes which have masses of several thousands to millions of Daltons have been discovered, but they can also carry out transformations of extremely small molecules such as carbon dioxide or even nitrogen. Okay. So, here is an example of carbonic anhydrase, which has a molecular weight of 31,000 Dalton, but catalyzes the reaction of breaking of or hydrolysis of carbon dioxide to bicarbonate or carbonic acid. Okay. Each enzyme molecule can catalyze the hydration of 1.4 million molecules of CO2. Okay. So, it is a huge number and the efficiency is extremely high, but keep in mind that the molecular weight of this protein is 31,000. Okay. So, the catalyzed reaction is about 10 power 8 times faster than the uncatalyzed reaction. Okay. And this rate enhancement is actually on the lower side for a typical enzyme catalyzed process. In order to understand how this catalysis occurs, let us look at a typical free energy diagram. So, here is on the y axis a free energy, and here is the reaction coordinate, and this is a starting material which is represented here, and here is the product. Okay. This difference in energy is delta G for the reaction, and here is what we call as activation energy or the barrier required for the reaction to occur. Okay. The general understanding of how enzymes function is that they lower the barrier or reduce the energy required for the starting material to go to the product. And so, therefore, enzymes create a new transition state which is lower in energy compared to the original transition state for conversion of a starting material to a product. Okay. Now, as we can imagine that an enzyme must have a site at which the substrate must bind. Okay. This is called the active site. Now, this site can be a groove, a hollow, a gully, or any of these small structures where the substrate can actually get in. Right. And once the substrate gets in, there is a binding that occurs, and this binding results in a change in the conformation, and of course, the enzyme can now do whatever it is supposed to do, and then finally, the product is released. Now, this binding site or the active site provides a suitable environment for many difficult to carry out reactions. And not just that, some of these reactions are not even possible in aqueous environment, and as we discussed earlier, the hydrophobic environment that is produced in this active site is responsible for binding, and subsequently we will look at some of the residues that can help with catalysis, but binding is an important part of enzyme catalysis. So, the active site contains several amino acids which play an important role in catalysis. Okay. Now, one can derive some information about how well the active site is conserved across various organisms evolutionarily by comparing the sequence or the primary structure of enzymes from various organisms. Okay. Now, it is possible that the primary structure differs from species to species, because this primary structure has gone through a number of iterations or mutations happening over millions of years. Okay. The variability in this is somewhat proportional to how far apart these organisms are on the evolutionary ladder. So, for example, something that is evolutionarily in a early phase can have very different primary structure from some enzyme which is in humans, right. But there are amino acids which remain constant through evolution, and these can be crucial to the enzyme's function. Okay. And these amino acids can often present themselves in the active site, right. Now, it is also possible that once the active site has evolved, it is also possible that it can be altered through mutation. Now, if the mutation is not useful, then the enzyme can lose its function, and the cells which lose the function can have a poor chance of survival. And so, therefore, there are enzymes which are very crucial to the functioning of a cell, wherein even a small change in a particular amino acid can have a dramatic impact on survival. Now, it is also possible that this mutation would therefore not be preserved unless it of course improves the substrate binding. So, to sum it up, there are two major roles of amino acid in the active site. The first one is to bind the substrate, or we will look at shortly, it has to bind a cofactor, and the second part is in catalysis, where the transition state energy is lowered, right. And so, both these processes are very crucial for an enzyme to function. The third part, which is basically dissociation, is also important, because if the product binds very strongly to the enzyme, then the catalysis, the turnover is not very good, and the catalyst is not going to function very well. Okay. Now, as we looked at earlier, the binding interactions between the substrate and the enzyme are largely non-covalent in nature, and these non-covalent interactions we have already classified them as ionic bonds, hydrogen bonds, dipole-dipole or ion dipole interactions, and hydrophobic interactions and van der Waals interactions. Okay. So, one of the examples that we can look at is the conversion of pyruvic acid to lactic acid by this enzyme called as lactate dehydrogenase. So, here it uses NADH as a reducing agent, and this ketone is converted to an alcohol. If we look at the kind of interactions that the substrate would have with the protein, then you would see that there is a methyl group which can involve itself in hydrophobic interactions or van der Waals interactions, and there is a hydrogen bonding capability. All these carbonyl carbons and the O minus can involve themselves in hydrogen bonding, and of course, there is an ionic interaction here in the carboxylate, right. So, if we have to divide this molecule into various modes of binding, then all these three interactions must play or could play an important role in order for effective binding to take place. Okay. Now, therefore, the knowledge of how a substrate binds to the active site is really important, because if we want to inhibit that enzyme, or if we want to design a drug that can inhibit that particular target enzyme. So, let us understand how the enzyme catalyzed reactions occur. The first major hypothesis that was presented was the lock and key hypothesis. Now, the lock and key hypothesis is a very simple mechanism. So, you have a lock which has a particular groove in it, which is very selective to a key, which is the substrate. And now, once the key presents itself to the lock, it binds and forms a very rigid binding, and therefore, then you can have the catalysis occurring and the dissociation of the product. Although this is a very interesting and important hypothesis, it does not really explain how some enzymes can catalyze a range of reactions. So, if you have a lock that is very specific to a particular key, then how is it that we are able to open the lock with a number of different types of keys? So, therefore, the lock and key analogy is not exactly something that is very optimum for us to understand enzyme catalysis. The next set of hypothesis that came up was the induced fit hypothesis. Okay. So, here the enzyme binds to the substrate, but during the binding the shape of the enzyme changes, and this is some sort of a molding process which then results in effective binding. For example, you have pyruvate here which is sitting over here, and now once pyruvate binds to or starts to bind to the active site, there are conformational changes which can occur which help with improved binding of certain areas of the substrate with certain areas of the active site. So, this helps us understand why an enzyme can catalyze reactions with a number of substrates which do not necessarily have the same structural features. Now, each substrate changes the active site into a shape that is ideal for it. Okay. So, as long as the molding process does not distort the active site such that the mechanism does not occur, the substrate would be turned over. Okay. Now, it is obvious that the substrate itself is not a passive spectator, and it also undergoes certain rotations and conformational changes which help with improving binding interactions. Okay. Right. So, therefore, the enzyme changes shape to maximize the binding interactions, and the same thing can also happen with the substrate. So, this hypothesis, which is the induced fit hypothesis, helps us understand how an enzyme functions and how an enzyme can actually catalyze a range of reactions with varied substrates. And from the substrate standpoint, when you have a diversity of substrates, since there is an induced fit that occurs, the binding can help weaken certain bonds, it can stretch certain bonds, and it can strengthen other bonds, and therefore, this interaction can be useful in accelerating the transformation that can occur. So, the enzyme undergoes a conformational change, it starts to change its shape. The substrate can also undergo a conformational change, and if this conformational change results in stretching or weakening of bonds that need to be broken, one can explain how the transition state goes down in energy. It is also possible that maximizing these bonding interactions may force the substrate into the ideal conformation where the reaction can occur in a very facile manner. Okay. Once it is bound, the substrate has to undergo a reaction. It is almost like how you fix the victim which cannot who cannot evade the attack, and once you have weakened the defenses, which is by weakening bonds or stretching bonds, the reaction is easier because the activation energy is lower. As I mentioned earlier, these binding interactions with the active site have to be strong, but they cannot be so strong that they do not let go after the reaction is done. If the product is stuck, that does not dissociate, it would basically block the active site from catalyzing another reaction. Therefore, it is important that a balance be struck here, and you would want adequate binding and adequate dissociation for a reaction to be effective. Okay. Now, let us look at what a transition state is. A transition state, as we looked at earlier, is a high energy situation between the starting material and product. And now, since we have suggested that the enzyme reduces the transition state energy, the reaction goes faster when compared to the non-catalyzed reactions. Okay. One of the ways in which so histidine is important because histidine is a source of proton, and since water molecules are not normally present inside the protein, histidine can act as a source of acid or base, and therefore, an important way to carry out acid base catalysis. Okay. There are other amino acids such as glutamic acid, aspartic acid, or even tyrosine, which can donate protons, but this is one way. So, therefore, it can donate protons, and therefore, it can act as an acid. The other important way in which catalysis occurs is through the use of nucleophilic groups. Okay. One of the best nucleophiles inside the cell contains a thiol, which is a cysteine residue, and the pKa of thiols are typically in the range of 7 to 9, but they can go down depending on the local environment. Thiolates are excellent nucleophiles, and therefore, they can act as a nucleophile in certain reactions. Serine, on the other hand, contains an alcohol, and alcohols, as we know, are not very good nucleophiles. However, when a histidine residue is present right next to a serine, it is possible that the histidine can pick up the proton from serine during a nucleophilic attack. So, here is an example of an amide being hydrolyzed by a serine residue. Okay. So, if you look at this example of chymotrypsin, so here a serine residue can donate a pair of electrons, or it can attack the carbonyl, and subsequently once it forms a protonated oxonium species, the histidine is present right next to it to pull out the proton and quickly remove the proton from the oxonium species. And subsequently, the tetrahedral intermediate, which has an O minus, can pick up a proton from histidine, and therefore, histidine can act as a acid base catalyst in this case. Okay. So, this is an example of how these three amino acids, serine, histidine and aspartate, act as a catalytic triad.